Welcome students. Today we are going to learn about light and photosynthesis. And in the previous class we discussed about air and photosynthesis, water and photosynthesis. In this session we are going to discuss about light and photosynthesis. Okay. Here, in the time of Joseph Priestley, the scientists don't know about the relationship between light and photosynthesis and the concept of energy. But in the later time, many scientists they conducted experiments on this to reveal the relationship between light and photosynthesis. Okay, here to know about the relationship between light and photosynthesis, first the scientists started hypothesis in this manner. Whenever oxygen molecule added to a molecule of carbon, it just produces carbon dioxide. Whenever oxygen molecule adds to hydrogen and it just forms water molecule. Okay? By the formation of carbon dioxide and water, along with these two, energy is releasing in the form of heat. If you take a burning reaction, if you take any kind of burning process, in that reaction carbon dioxide is releasing and water vapor is releasing, along with these two, energy also releasing in the form of heat. We all know. What about the opposite reaction? If we take these two, carbon dioxide and water, what will happen? Carbon dioxide and water, whenever these two combines, it forms glucose. We all know. It forms glucose or carbohydrate. Along with that, water molecule and oxygen is liberating into the air. Okay, that is the opposite of this reaction. If we talk about the concept of energy, for the production of oxygen by green plants, that process also requires energy. Where does this energy come to the plants? How the plants able to get energy in order to produce oxygen? That is the question now. And for that question, answer is discovered by a scientist called as John Ingenhousz. John Ingenhousz. He is a Dutch scientist and he conducted his experiments by taking a Hydrilla plant. We all know that it is a aquatic plant. Okay, any kind of aquatic plant you may take, and we are able to conduct the experiment like John Ingenhousz. For his experiment, what he took? A Hydrilla plant, which is aquatic plant. By taking that Hydrilla plant, he started the experiment. Whenever he exposed the Hydrilla plant in the bright sunlight, then he finds some gas is releasing into the water in the form of bubbles. Later he identified that gas as oxygen. He identified that gas as oxygen. Okay, that was revealed by the scientist John Ingenhousz in the year 1779. In the year 1779, what he discovered? The role of light. If there is no light, there is no photosynthesis. That what he revealed. After that, this experiment is also further confirmed by another scientist in the 20th century. He is Engelmann. The scientist Engelmann confirmed the result of John Ingenhousz. What is that? Sunlight is necessary for photosynthesis. If there is no sunlight, then what will happen? There is no photosynthesis and there is no liberation of oxygen. There is no production of food. That what revealed by these two scientists. And he proved that experiment by taking a strand of algae. He took a strand of algae and he exposed this strand of algae to different colors of light. Different colors of lights in the sun, we all know rainbow. A rainbow, it contains different colors like Vibza, you already studied in your earlier classes, different color lights. He just exposed the strand of algae to different colors of light, and he identified whenever he exposed different colors of light, then oxygen sensitive bacteria, it just crowded to the light where it is exposing to the blue region and red region. Okay, the blue region and the red region, blue light range, where the, and he also detected that the point of maximum photosynthesis, the rate of maximum photosynthesis, where does it occurs, at which wavelength of light it occurs. This was discovered by Engelmann in the 20th century. Okay, here the blue light, which is having the range of 425 to 450 wavelength, 425 to 450 wavelength, and at the red region also the rate of photosynthesis is maximum. What is the wavelength of that red light? That is about 600 to 700 nanometers, 600 to 700 nanometers. Okay, the plants are observing the sunlight by the help of some colored pigments present inside the plant, and that colored pigments we are calling, generally we are calling as chlorophyll. The green color pigment present in the plant we are calling as chlorophyll. Means this chlorophyll able to trap the sunlight very easily. Okay, and he also give the concept of pigments, different colors of pigments present in the plant. Mainly here the chlorophyll absorbs this energy from the sunlight. That energy is very useful in the photosynthesis process for the production of oxygen. I think you understand this introduction of a relation between light and photosynthesis, and what experiments prove that oxygen is produced during the process of photosynthesis in the presence of sunlight. We all know that in the process of photosynthesis, the green plants able to produce oxygen. Is it right? Yes. The plants are able to produce oxygen. Where they are able to produce oxygen? In the presence of light, sunlight. If there is no light, then there is no photosynthesis. Then now we are going to discuss about a small experiment which is called as Hydrilla experiment. By taking aquatic plant like Hydrilla, we are doing this experiment. Then what is the aim of this experiment? To prove oxygen is produced. Here you have to remember the word produced. Produced during photosynthesis in the presence of light. Then what are the requirements for this? You need a beaker, a funnel, water, test tube, and at the same time you also need fresh Hydrilla plant. If there is no Hydrilla plant available to you, you may take another aquatic plant called as Alodia. Alodia plants also you may take if the Hydrilla plant is not available. Okay, those are the requirements for this experiment. If we talk about procedure, for this in the procedure, first you take a beaker and fill the beaker with water. Fill the beaker with water. Now take a short stemmed funnel. The stem of the funnel should be short. Such kind of funnel you have to take. Now insert the collected fresh Hydrilla plant into the funnel. By inserting the Hydrilla plant into the funnel, you just keep the funnel into the beaker which is having water. Okay? Upside down in a inverted position. Okay? The mouth of the funnel should be to the bottom of the beaker. Later, you have to take a test tube and fill the test tube with water. Fill the test tube with water and carefully you should insert the funnel into the stem, the stack of the funnel. Okay, like this. How like the operator set here, like that we have to arrange all the necessary things in this manner. Now prepare 2 sets of operators and keep one operators in the sunlight, another operators in a dark room. One operators in the sunlight, another operators in the dark room. After keeping the arrangement, the full setup, about 2 to 3 hours, you may find some air bubbles or arising into the test tube. And you also notice another point here, the test tube is going, it just looking like somewhat region, the upper portion of the test tube, means the bottom of the test tube is becoming empty for a long time. If you keep for 2 to 3 days, most of the water they may enter into the beaker and the test tube it just looks like a empty one. Okay, like that here some gas is collected near to the bottom of the test tube, and whenever we keep this operators in the sunlight, here you may find bubbles. What are these bubbles? Yes, what you are thinking is right. These bubbles are oxygen bubbles, means a gas of oxygen is liberating from this Hydrilla plant and it is collecting at the bottom of the test tube. Now we have to test whether the gas collected in the test tube oxygen or not. For this, you have to remove the test tube carefully from the beaker by closing the mouth of the test tube with your thumb. By using your thumb, you have to remove the test tube carefully without unnecessary shaking. Okay, and you should not give any space to escape of the gas. Now take a matchstick or a incense stick, what is available, and keep near to the mouth of the test tube, then it may burst like a flame. We all know that oxygen, it always encourages the process of burning. That's why the flame becomes bigger. It blasts like the matchstick, it blasts like a flame. Okay. By this experiment, we can prove that oxygen is produced during the process of photosynthesis. What is the result? Oxygen is produced during the process of photosynthesis in the presence of sunlight. What about the operators which we kept in the dark room? There is no air bubbles. There is no air bubbles because the operators present in the dark room and there is no availability of the sunlight. That's why the Hydrilla plant unable to follow the process of photosynthesis. Then it leads to the absence of oxygen in the test tube. Okay. Then now we are going to discuss about another small experiment and which is also important experiment. This experiment is also called as Hydrilla experiment. Now we are going to discuss about another experiment. It is called as black pepper experiment. Black pepper experiment. Why we are calling this one as black pepper experiment? Because in this experiment we are using a black pepper. Then what is the concept of this experiment? Sunlight is necessary to the formation of starch in green plants. Sunlight is necessary for photosynthesis in a simple way. Sunlight is necessary for the process of photosynthesis. For this process, what is the aim of this process? To prove that sunlight is necessary for photosynthesis. What are the operators we have to take? A potted destarched plant. We need a potted destarched plant. Okay, along with that we also need a black pepper or a silver foil, whatever it is. You may take which one is available, that one. And a scissor to cut the pepper into a design. Along with that we also need a petri dish and iodine solution, tincture iodine solution. Okay. If we talk about the procedure. In the procedure, first we have to take a potted plant and we have to keep the potted plant in a dark room for destarching the plant. We all know how to destarch the plant. We have to keep the plant in the dark room. Okay, we have to keep the plant in the dark room about 1 week. Then all the starch present in the leaves is used up by the plant. As it is present in the dark room because of the absence of sunlight, it is unable to do the process of photosynthesis. That's why it is free from starch. The leaves are free from starch. Now we have to take that destarched plant and we have to keep the setup in the sunlight. Okay, whenever we keep in the sunlight, you take one black pepper and cut the black pepper in the middle, what the shape you like most. Okay, either yes or A, B, any kind of shape we have to cut it. And you place that black pepper on the surface of the leaf by the help of some clips. By using clips, you just hold the black pepper on the surface of a leaf and allow the operators about 3 to 4 hours in the bright sunlight. After that, you just separate the leaf from the plant and place the, later we have to follow the process. First what we discussed, we have to remove the chlorophyll from the leaf by boiling the leaf in methylated spirit by using water bath. Okay. Later, you just spread the leaf on a petri dish. With the help of a brush, you have to remove the leaf and you have to spread the leaf on a petri dish. Now add 2 to 3 drops of iodine solution, tincture iodine solution. You just spread throughout the leaf. After a few minutes, you find that some portion of the leaf turn blue-black and remaining portion of the leaf, it just remains in the same color, green color or pale in color. It just remains the same. It indicates that the region of the leaf which is exposed to the sun, the region, which region is exposing to the sun? This part is exposing to the sun, and in the middle part what we cut into different shape, that part also exposing to the sun. That's why whenever we are doing the iodine test, then in that test we get the result, the shape what we cut turns into blue-black color and the bottom of the leaf and the front region which is not covered with black pepper also turns into blue-black. But the region where the leaf is covered by black pepper does not changes its color. By this experiment, we are able to prove that sunlight is necessary for photosynthesis. Okay, do you understand, children? And we will meet you in the next session with another topic.